I think you have joined the right, Microsoft team. Those who are in some other, connection, please come back to the normal one. I'm just going to, okay, 21 have joined. So let me start the class. First, let me share the screen. So is the screen visible?
Yes, sir.
Okay, I'm putting it in the, uh, okay. So let us see. Yesterday we have talked about the carbohydrates which are linked with the proteins, and today we will talk about some specific function of these carbohydrates. To very quickly recapitulate, you have learned that the carbohydrates can bind with the proteins, and this kind of a binding is occurring with their N-terminal. And N-terminal glycosidic linkage can have a strong bonding with the protein. Now, this kind of structure of the protein, because they are linked with a carbohydrate, they are called as a glycoproteins. Now, these glycoproteins, they have a certain function. That is what we have talked about. Main function you have learned is that there is an interaction between cell and cell where these glycoproteins are involved. They can act as a receptor, they can act as a molecule for cell interaction, and we have given some of the examples. Then we have also talked about the very specific kind of carbohydrate binding protein. Kindly remember that this thing is different than the glycoproteins. Lectins are the protein which can bind with the carbohydrate, whereas the glycoproteins are the one where carbohydrate is bound already. These lectins, they also have a specific function which we have discussed in the previous class, and have given some of the specific examples. So these lectins, they are proteins and they can interact with the carbohydrate, and that's the way binding occurs. One of the very specific example I have taken was about the influenza binding protein, where the binding occurs through the sialic acid. Then what you have learned is specific carbohydrate which is present here. For example, in this protein, you can say that sialic acid binding sites are present. This is the protein. It has a binding site for sialic acid. And what is a sialic acid? Sialic acid is nothing but a carbohydrate. So a protein can bind with carbohydrate through a very specific carbohydrate moiety or carbohydrate unit, which is a sialic acid. This part was discussed in the previous class. And this sialic acid is a carbohydrate. What you have is that this is the structure of more or less the structure of glucose, but what you have additional is N-terminal is acylated. One of the H is carboxylic group, and you have two additional groups. So what you have is, instead of 6 carbon molecule, you have a 9 carbon molecule. Now let us move on to a very interesting concept. And here what we are going to talk about is that how this structure works and what is the important role. Are you able to hear?
Yes, sir.
Okay, fine. So here, the carbohydrates, they also participate in the molecular targeting. Now I'm coming back to the point that how the carbohydrate, they play an important role. First thing is that you must understand what is a molecular targeting. As name indicates, molecule is the one which you are interested in targeting to a specific organ, and that is what can be referred as a molecular targeting. So molecular targeting is, suppose I want that my protein should target to liver, I should be able to do that. So in this case where we are talking about the role of a carbohydrate, let us talk how some of the protein, you can see that there are proteins like immunoglobulin. What are immunoglobulins? Immunoglobulins are a protein which provides us a defense. I will talk about protein a little later, but this is the one which is nothing but antibodies. In general, in a layman's language, we call them as antibodies, and in a scientific language, we call them as a immunoglobulin. So it's a globular protein and responsible for immunology or immune response, and that is why they are termed as immunoglobulin. So many immunoglobulins, they are synthesized. How they are synthesized? They are in the response of any foreign antibody. These immunoglobulins are synthesized. Then newly synthesized peptide hormones. There is another category. These hormones, which are peptidic in nature, proteinaceous in nature, they are also present in our blood circulation. So they have their own function. So these immunoglobulins and peptide hormones, they have their own function. They are synthesized inside the body. And what happens that it contains a carbohydrate unit. Tell me how they will have a carbohydrate unit, how they can be considered. These are proteins. They have a carbohydrate unit. So what does it mean? It means that all these proteins, they are, what should we name them? They should be glycoproteins because they are linked with the carbohydrate. Now, there is a very specific nature was found in these proteins that they have a sialic acid residue. And I have just now discussed what is the sialic acid. So there is the sialic acid, which I am talking about. This is the one which I was referring to. And then because the sialic acid, so sialic acid alone will not be present. Sialic acid, I will tell you how they are present. So sialic acid is the one which I have shown you earlier. I'm just going back to a couple of slides. And here I have shown you this one where, sorry, I'm just trying to take a pen and show you that this is the one which is sialic acid. Now what you have is that here you have a protein. This is the one which is asparagine. So rest part, so there is a protein. Now this protein has this carbohydrate. We term as them as a, we call them as a glycoprotein. And since sialic acid is present in the terminal, we refer them as a sialoglycoproteins. Is it clear to all of you?
Yes,
So
sir.
sialoglycoproteins are proteins which has specifically sialic acid in their terminal, and that's why they have been referred as a sialoglycoprotein. Okay, so I hope that this concept is clear to you, that there are many proteins, glycoproteins, and if I look at the function of this glycoprotein, it could be variable. It could be that they are acting as immunoglobulin. It could be that they are when I'm saying this, what I mean to say here is that the function of these glycoprotein could be like antibody. The function could be like peptide hormone. But all of them, they have in terminal position a sialic acid residue. And that is why we call them as a sialoglycoprotein. So these sialoglycoproteins, as I have shown in previous slide, has a core sugar, glucanag. What is a glucanag? Is an N-acetyl glucosamine, and then galactose and sialic acid. So this sialoglycoprotein, this protein could be variable. Protein could be immunoglobulin. Protein could be peptide hormone. But the carbohydrate, which is a link at the terminal, is common one. Is the one which has at the terminal sialic acid. Now what happens? Suppose the sialoglycoprotein is present. Now your immunoglobulin has synthesized. It has interacted with the say antigen. After interacting with the antigen, over. Answer is that this protein should be finally degraded, should be removed from the circulation. Otherwise, what it is doing is that it is, consuming our protein. It is also circulating unnecessarily in the body. So the main thing here is that either immunoglobulin or peptide hormone, if their function is over, they should be removed circulation. It's done. Now, one concept is that something has been synthesized and it has to be removed. Another concept is that these molecules, they have been designed, they have been, engineered in such a way that at the terminal there is a sialic acid. Now, look at the mechanism, how it happens, why it should happen, right? So, sialic acid is the one, as long as sialic acid is present in these protein. Now, I will refer these immunoglobulin and peptide hormone as a sialoglycoprotein because they have a sialic acid in their terminal. Now, what happens? They keep on circulating, and once their job is over, this enzyme known as a sialylase enzyme, it is present in the blood capillaries, and what it does is that it cuts it. It removes sialic acid. So enzyme, which is a catalyst, what it will do is that as long as they are functioning, it will keep on circulating. As soon as their job is over, their function is over, what they will do is that they will remove the sialic acid. This part will be cut. Now, if I remove sialic acid, what is left out in the protein? And what should we name them? They will be without sialic acid. So you have a protein, sialoglycoprotein, without sialic acid. So if I have a protein, sialoglycoprotein, without acid, we name them as a sialoglycoproteins. So once you cut this bond, which is between galactose and sialic acid, two things are happening. One thing is the sialic acid is removed, and second thing is that galactose is exposed. Now, what happens that liver cell, so suppose I have a liver, and here you have a sialoglycoprotein, this sialoglycoprotein, once as long as sialoglycoprotein is present, they will not be recognized by the liver cell. So suppose I have a liver cell. So this is a liver. It has a lot of receptor, and these receptor, do you understand what is a receptor? Receptor is the one which can bind with a small molecule like ligand. So you have a blood where sialoglycoprotein is circulating, and what has happened that sialoglycoprotein, as long as it is circulating, it goes to the liver, it will be rejected, it will not bind. But as soon as the sialic acid part has been removed, what will happen? Here you have a galactose, here sialic acid will be removed. What you will have is, you will have a galactose. Now this galactose, I'm not able to use my pen correctly. So what will happen, that galactose will be the one which is present. And as soon as galactose is there, it will bind with the receptor present in the liver cell. So asialoglycoprotein, they bind to the liver cell because can you name this receptor? This receptor is known as asialoglycoprotein receptor. Very simple thing. So liver cell has got asialoglycoprotein receptors, and they will bind to asialoglycoprotein once they are formed in the blood capillaries and then reach to the liver cell, and then they will be recognized. And after recognition, what will happen? These asialoglycoprotein, as soon as they bind with the receptor, they are internalized. Internalized means they are taken inside the cell, and the phenomenon is called as endocytosis. And because of this endocytosis, the asialoglycoprotein is removed. So what has happened? The immunoglobulin or the peptide hormone, which was chemically, which was in the form of sialoglycoprotein, has been removed from the circulation because that has been recognized by the liver cell that no, I do not have any function for these molecules, and that is where they have been removed. Once they are removed, what will happen? They will be circulated, new sialoglycoprotein will be synthesized, and they will be again doing their function and will be removed when their job is over. So this whole phenomenon is called as a molecular targeting. Tell me if it is clear to you.
Yes,
I am, for a second, I'm ending the show. Now tell me, anyone, any comment on this? Yeah, please let me know if you have any comment on this. Have you understood? The question here is that why, why the sialoglycoproteins should be present and what is their function or what is the molecular targeting. So it's very clear. Molecular targeting is where you want to target a molecule to a particular organ. And what you have done here is that you have a targeted sialoglycoprotein to the liver cell. And how it happened? It happened through the tailoring the terminal residue of sialoglycoprotein. So terminal residue was chopped off, and because of chopping off of this terminal residue, you were able to convert sialoglycoprotein into asialoglycoproteins. There was a receptor present already on the liver cell which can recognize these asialoglycoprotein, and once it is recognized, they were internalized and they were again sent back to the circulation for new synthesis of sialoglycoproteins. Okay, I have repeated the concept. It's a very interesting concept, and it gives you an idea that how important carbohydrates are, how they are, giving you a important function inside the body. With this, I have covered the carbohydrate, and what you have learned at the end is that you have learned that the carbohydrates are important biological molecules. You have also learned that carbohydrates are the question here asked is, are there other examples of molecular targeting? I think that any molecule which has a sialic acid on the terminal, they will lead to the molecular targeting. And this is because liver cell is the one which is important factory. Here, what happens, that detoxification occurs. This is the part of the cell body where most of the detoxification reaction occurs, and that is why the sialoglycoprotein receptor is there, and that's the way it is being recognized. Other than that, you have already learned that these carbohydrates which are present on the surface, they are going to be involved in the interaction. Now, I was recapitulating what all you have learned. So we have talked about the carbohydrate, important biological molecule, learning about their functions, which are important in the sense that individual carbohydrate or individual saccharides like glucose, like fructose, like galactose, all of them, they are important in their function. Now, you have learned that what is the very important function of galactose? I need an answer from you. The question here asked is, after returning from the liver, only sialic acid attaches for formation of sialoglycoprotein. Answer is yes, if they have to be synthesized, if it is the same molecule, because the protein, it depends on which protein we are talking about. Is it a peptide hormone? Is it a immunoglobulin? All of them, they will be resynthesized. Otherwise, this can be further degraded and this protein can function in a different way. It is not necessary that always sialic acid attaches and it has to work. So what I'm saying here is this is a recycling mechanism where on one hand it can recycle, on the other hand, if it is not required by the body, then something else can be synthesized. So proteins, they keep on there, they have a life. Slowly you will be learning that each protein has its life, and once their function is over, they can be recycled to individual amino acid, and they are involved in the synthesis of different proteins. So that part I'm not talking about. That is coming under how proteins are degraded or how proteins are destined to be degraded. That's a different thing. That's a different thing which you learn in the cell biology. I'm currently focusing on the carbohydrate linked proteins. Okay, so now we are discussing about the general understanding in this whole carbohydrate chapter is that you have learned about the smaller structure, which is a monosaccharide. Important monosaccharide you have learned are glucose, fructose, galactose. I had asked a question that what is the role of a galactose? I said that galactose is an important one. Glucose is important because it can be further metabolized. You will be learning the metabolism. You will be learning how glucose can produce energy. What is the role of a galactose? Galactose can also enter in the same metabolic pathway and can do the job. But what could be the specific function of the galactose? Galactose can be involved. I'm not getting answer from you. Just now we have discussed about it. Galactose is present in a terminal part after the sialoglycoprotein and can be useful for recognition of the protein. So it's a very important component of oligosaccharide, which is a sialoglycoproteins. So now we have a couple of questions to be discussed, and then we will move on to the next chapter, where I think today I can give you introduction. But as the learning is concerned, the learning objective was about the monosaccharide, you learn about the oligosaccharide, you learn about the polysaccharide. So what you have learned about the monosaccharide is that monosaccharides are formed from polysaccharide by degradation. So suppose I have starch, or suppose I have a glycogen, they will be cleaved, they will be chopped off, you will get the glucose molecule, and they will be utilized for energy production. On the other hand, if I look at the function of polysaccharide, they are a storage molecule. And I have not discussed in a detail about why polysaccharide is a storage molecule, but still it is not stored in the larger quantity. Can anybody answer why polysaccharide cannot be stored inside the cell in a larger quantity? I have raised this question on the first day or second day, where we have discussed about the fact that though our 90% component in the food or in our diet is carbohydrate, at least 70%, what I have in storage is only 1% stored in the liver or the muscles. So answer here is the carbohydrates are quick source of energy, but it cannot be stored in a larger quantity in the cell. Why it is so? If you look at the structure of a carbohydrate, most of them, they have, okay, somebody wrote about the osmotic pressure in the cell. The answer, or this answer is correct when we are looking at the individual carbohydrate, when we are looking at the monosaccharide component. So you cannot store large amount of monosaccharide, because that will create osmotic pressure and that will burst the cell. This part we have already discussed. My question is slightly different. My question is why the polysaccharide cannot be stored, because polysaccharide will not produce that much osmotic pressure. But why polysaccharides, for example, why don't we have all the energy requirement stored in the form of, say, glycogen in animals and as a starch in plants? Why we have to resort to convert carbohydrate into lipids? Okay, you have to look at the structure. If you look at the structure of a carbohydrate, you have a several OH group that is given integrated in there itself. Now, when you have a several OH group, what they do? They bind or they interact with the water. So they require larger space to be stored. And if I compare it with the lipids, what are the lipids? I will give you the structure of lipid when we talk about the lipid. But in general, you know that most of the lipid which is stored in our body is in the form of triacylglycerol. That means you have a glycerol structure, and each moiety is attached with the fatty acids. So the function is that if triacylglycerol to produce energy, it will be broken down into glycerol and fatty acids, and the fatty acid is the one which is going to interact. But now if you look at the glycerol structure, in the glycerol structure, it is hydrophilic or hydrophobic? CH2 group cannot interact with the water. On the same way, your fatty, because if fatty acid is free, it can interact with the water. But if fatty acid is a bound to glycerol moiety, that means acylglycerols, they cannot interact with the water. So triacylglycerol molecule, they are hydrophobic in nature, whereas the carbohydrates, they are hydrophilic in nature. So if I have a hydrophilic molecule, lot of water molecule they will accumulate around this. So you will have a hydrophilic interaction. On the other hand, if I have a lipid-like molecule, they will be hydrophobic in nature. They will be requiring very little space. Initially, we have discussed that because water loves another water, so water-water interaction will force them to come and make a bigger droplet. So you will have triglycerol stored in the form of a bigger droplet. Now, look at the chemistry and very general understanding. If you know, if I compare a carbohydrate with a lipid. Now, what is the structure of a carbohydrate? If I give the molecular formula, what is this general formula for a carbohydrate? It's a 6CnH2O. That means you have as many oxygen as many as a carbon. So if I compare two 6 carbon containing molecules, one carbohydrate, one lipid, what you will find? The carbohydrate will have more oxygen compared to lipids. So now at least this answer I want from you, write on a chat that which one is a more reduced form? Carbohydrates or lipids? Which one has a less oxygen? So whether I have a 2 carbon atom or 6 carbon atom or 20 carbon atom, the lipid molecule or the fatty acid will have only 2 oxygen. But in case of a carbohydrate, depending on the number of a carbon atom, your oxygen atoms are being decided. So lipids, they are the more reduced forms. Answer written by Depriya is correct that lipids, now in lipids, we are comparing the fatty acid. So anything which is in a more reduced form that has a tendency to be oxidized. And if something can be oxidized, then it becomes more energy producer inside the cell. Okay, this concept is a very general concept which tells us that on one hand, lipids can require lesser space to be stored compared to carbohydrate. On the other hand, these molecules have more potential, lipids have more potential to produce energy when they are oxidized because all the energy produced in the cell is by the process of oxidation. Is this concept clear to you? Okay, next question is that if you look at the protein structure, this question I'm framing today, we will discuss it at the later stage when we talk about the other molecules. But the question here is that polysaccharides, their number, for example, if I ask you what is the number of residues or number of monosaccharide in starch, you cannot answer. Nobody can answer because it depends what is the source of starch. It could be 1000 glucose molecule, it could be 10,000 glucose molecules. So there is a range which is always given in the polysaccharide. So polysaccharide will have units and there is a range which is decided. So I can say there are 1000 unit of glucose present in starch obtained from such and such source. So the starch which is obtained from say rice or starch which is obtained from our say potato may have a different amount of the monomeric residues. On the other hand, this number of residue also depend on the stage. So suppose I get something at a particular stage, they will have a different number of carbohydrate or number of monomeric unit than from the stage. I'm talking about the growth stage or accumulation stage. Okay, so is this concept clear? In other words, we can say that polysaccharide does not have the exact number of a monomeric unit. Is it clear to you? Now, if I look at the protein, protein will always have a particular number fixed in their structure. Suppose I say it is 100 amino acid, it will always have 100 amino acid. Same way, if I talk about the nucleic acid, if I say there are 25 nucleotides are present, there are 25 nucleotides will be present. So that way the numbers are fixed. Whereas in this case, in case of a polysaccharide, numbers are not fixed. Why it is so? Why polysaccharides are acting as a bin of energy? In other words, we are talking about that suppose I have some energy which is stored, some extra glucose is there that will be dumped in a bin so that a polysaccharides are formed. So think about it. We will discuss in whenever we talk about the other molecule and their functionality. On the other hand, you have also learned that the oligosaccharides, they are not like polysaccharide. They are different from polysaccharide in the sense that if I have a sialoglycoprotein, if I change one of the monomeric unit at the terminal end, their function will change. On one hand, it is circulating. On the other hand, as soon as I remove the sialic acid, that becomes important. So this is a very interesting function, and it's important for oligosaccharide to provide a function because they are destined to do some activity based on their structure. Further, I would like to give you one more interesting concept about the carbohydrates. So, when we are looking at the carbohydrate, we are looking at them as an energy source. We have also looked at their specific function, where we have talked about polysaccharides. When we are looking at the monosaccharide, we have looked at their individual energy function, but you have seen that most of the monomeric unit, they enter in the energy production pathway, which we will discuss during the metabolism, through the same route, whether it is a glucose, galactose, fructose, though their priority may be different because of the presence of enzyme, but all of them, they metabolize in more or less in the same way. Okay, I will not talk about the metabolism, etc. So let us move on. After completing this, I'll just have some time to introduce you to a very simple concept about the proteins. And we'll just talk about the structure and function of proteins. Can you see this PPT? Is it visible?
Yes, sir.
Yes, sir.
Okay, it
Yes,
takes
sir.
time. Probably it is taking time, no problem. So now next chapter which we are going to talk about is proteins, structure and function. So you have learned about the one of the major macromolecule about their structure and function. Now we are going to move on to the second molecule, which is the proteins. We are going to talk about their structure and function. Proteins are most versatile macromolecule. Why I'm talking about versatile molecule? In a few minutes, you will learn that what is versatility in these molecules. So if I look at these molecules, they have several functions, and these functions, they are ranging from their function as a, so they have a lot of crucial function, and if the proteins are not present, these functions are not going to be accomplished inside the cell or inside the body. So what are these functions? So one of the interesting function which I am going to talk about is a catalytic function, is a function where the the proteins, they are going to act as a catalyst. What is this? You have already learned about the catalyst. You have learned about the inorganic catalyst. Here you will be learning the catalyst, which is a protein based catalyst. So we will have a full chapter on catalyst. We call them as enzymes. I will define why we call them as enzymes, but in general we call them as a biological catalyst. So we'll be learning about the biological catalyst, and more or less proteins are the major biological catalyst. There was a time when people, they said that the proteins are the only one which only one type of molecule which can act as a catalyst, but later on found that there are another type of catalyst is also present, which is RNA based catalyst. So they were named as a ribozymes, but we are focusing right now about the protein based catalyst. So for example, one of the very simple function which is being catalyzed and because of which we are surviving is carbonic anhydrase. This is the enzyme. What it does is it has a very simple process. What it does is that it converts bicarbonate into carbon dioxide and water. Now this reaction is a very, very slow reaction, and in presence of a catalyst, it is a very fast. Can you tell me where in our body this type of reaction is important? Any one of you? Can you think of a reaction? Any one of you can,
Sir,
you
can you
know,
please repeat?
see, I have said that there is a reaction which is being catalyzed by carbonic anhydrase, and what it does, it converts carbonic acid, that is into water and carbon dioxide. Where do have this important reaction in our body? And if it doesn't happen, what will happen?
Sir, lungs.
Okay, that is correct. So it works in the lungs, and what it does, it converts carbonic acid into carbon dioxide, as it is a logical answer. But how does it, how it is important? It is just converting, carbonic acid into carbon dioxide, but how does it affect our cellular metabolism?
Sir, it is supposed to
Remember,
remove carbon dioxide dissolved in the blood, so to like maintain the acidity of the blood, I think. Sir, you're not audible.
is that it maintains Can you hear me?
Yes, sir.
Okay. Now I'm going to share the slide. For a few seconds, there was an issue in the sharing, and you know, I was unable to. I'm stopping the sharing and open the tray again. Very quickly, we will complete this part, and what I'm interested in is showing you that how this. Can you see the slide? I don't think that is PPT visible.
Yes, sir.
Okay, so what I have discussed is first function is a catalytic function, where I have taken one example, thousands of proteins which are acting as a catalyst, and all the reaction inside the body is dependent on these proteins because they are catalytic in nature. Then second thing is the transport and storage of other molecules like oxygen. So for example, hemoglobin, it transports oxygen in erythrocyte or in RBC, myoglobin that transports oxygen in muscle, and then you have other transport protein like transferrin, etc., which transports iron. So you have lot of a transport function. Now, they also provide mechanical support and immune protection. I will talk about that. Mechanical support, there are certain proteins which are acting as a, they help in the mechanical support. So instead of carbohydrate, here the mechanical support is provided by a protein. We don't have a polysaccharide like cellulose in our cell, and that is why the protection function or mechanical support has been taken by the proteins. And these proteins, they are, can you name any protein which is protecting our body? One thing is very clear, something which is in your hair or nails, they are keratin. There is something which is present in our connective tissue, that means the tissue which is just below our skin, these are collagen. So you have a collagen, you have elastin, and you have a keratin. These names I will give you, and I will give you the detail when we talk about the structure. But these proteins, they have a different structure, and because of a different structure, they can provide their function. What you have learned in the carbohydrate is that those carbohydrates, like cellulose, has a beta sheet structure, which is a more rigid structure, and because of that, they provide structural function. A similar concept will work in case of a protein as well, where the function of these proteins, they are because of their structure. So we will talk about that. Then they are also responsible for generating the movement in the cell, as well as you will see that they are involved in the transmission of nerve impulse. And example, can you name anything where nerve impulse is responsible for doing so? One is very clear. When you see something, how do you see something? You see because of the presence of a protein. So there is a protein called as a rhodopsin protein in our eye. and they act as a photoreceptor. So these are photoreceptor proteins in the retinal cell, and that's the way we see things. So that is one way, which is through the nerve transmission. Then you have a receptor proteins which can receive certain signal through acetylcholine, and that is where they can transfer the impulse. So this way they are involved in transmitting the nerve impulse. They also control growth and differentiation in the cell. So there are very many nerve factors, there are hormones, all of them, they are responsible for growth and differentiation. I will continue in the next class where we will be, discussing more about the protein, their structure and function, and just to give you some structure. These are the structure. We will be going into details of these structure. Now I have few minutes, 1 or 2 minutes. Any specific question you can raise? Any questions from you? If there is no more question, I will thank you, and please let me know where you have a difficulty in the class. I have recorded today's lecture. Let me save it. I will send it to you. Okay. Thank you.
Thank you, sir.